Yeah, my name is Jimin. So I come from the Center of the Microengineering Material, also the Department of Bioengineering, Bioengineering and Chemical Engineering. So I'm talking about the modular nanoparticle modified living mammalian cells and armored rebel cell. So our team is myself and Wei Zhu and Jacob Agola and Dr. Brinker. So we received several fundings for support of this one before, but right now we don't have a lot of support for this. And so for our staff, just quickly talking about what we can do is first we have cells, living cells, and then we coating with nanoparticle, and then we locking the nanoparticle onto the surface of the cells, make it not be internalized, make it not released from the cells, then coating to make like an armor on the surface of the living cells. So the reason why this become two different pattern is because of one is on the regular mammalian cell, they have the nucleus, they have all the things. Another one is the rebel cell. Rebel cell itself is a little different compared to the regular living cells because they don't have like a active uptake pathway. So it's more difficulty to modify the surface and also not easy to coating fully on the surface. But what we can, but for this one, so our nanoparticle coating to create this exoskeleton is just in seconds. It's super quick. So compared to the rest of the technique I will talking later, they typically like hours. So long time, not the more killing of the cells, living cells. So also another thing is because of we using the nanoparticle, we can using the mental organic framework particle, this three, and also, this four. And also we can using the silica, mispolar silica nanoparticle, and also the ion oxide, that magnetic nanoparticle. We can try different type, even the polymer one. We still try to optimize that one right now. And for this one, so since we coating on the surface of the living cells, we want to maintain their metabolic property. So all the living cells, they can still metabolic. Normally, they didn't show any very strange properties. And also we can, the rebel cell itself, for example, we coating on the rebel cell still can deliver the oxygen, still can passing through, have the long circulation properties. And the most important thing for this technique is we can enhance the resistance from the stress. So I will talk more details for this. So the background is because the mammalian cell, also the rebel cell is very delicate and very like easy to infect by the microenvironment stress. So when they have the like a detergent, ice crystal, toxic, or even some of the antibiotic, they will be recognized and kill the cells or something. But what we do is we just using the nanoparticle on the surface, just physically block it. So then we're thinking that we are helpful for the cell therapy, also because we're loading the nanoparticle onto the cells. So we're thinking also they can help for delivery the nanoparticles as well. And at the same time, since we have the nanoparticle on the surface, we can using nanoparticle itself as a sensor. So we can using to sensing the molecule release from the cells as well. So compared to the regular technique, typically people do the encapsulation of the cells. They do is layer by layer, polymer layer by layer, or phenolic acid deposition, or this is still polymer growth, different way, and also morph crystallization growth on the surface. All of them is typically time consuming, is like hours to do. And another thing is because they're directly using the source to prepare it. So if you want to have good structure on the surface, you're limited by the source of the biocompatibility. So a lot of things you cannot using. For example, ion oxide that nanoparticles, we can make it outside that need the high temperature, and then we're loading on surface. But for them, they cannot grow directly onto the cell surface by their technique. So I think we are the first one in the scientific area to coating the nanoparticle onto the cells. And also we can control the shell properties because the nanoparticle is already synthesized. So the nanoparticle pore size is already designed. So we know the porosity. But for all the rest of stuff, is typically out of control, just to make sure they can coating on surface. Also, we can choose in different nanoparticles. I will show we can have a multi-particle coating on the same cells, which is indicate we can do is a multifunctionalization of this, we call the armored cells or supercells, something like that. So the application is as I say, so this is the first thing they can like provide the protection from the strands. So the first thing we're thinking is they can using for the cell therapies because I will talking this later, but I will say this is because the nanoparticle itself can using be as a cell sensor, or we can deliver the cells because they can protect the cell when they inject to the in vivo that area. Also the rebel cell, the armored rebel cell itself, because they have the nanoparticle on the surface, they can be a super nano carrier to deliver the cells. You can see from these figures, this is from the chicken embryo wing. So you can say the armored rebel cell can circulate well, have no block inside that one. And so for the market, to our what we're thinking, as we say, so we physically block the surface of the cells. So it means if we inject the non-self cells into the some like animals or humans, they were not causing directly immune response at the first time because the nanoparticles, the exoskeleton, will block that like antibody, or they will block the any like a microphage interaction or dendritic cell interaction to the injection, the non-self cells. As you know, so this is a benefit for the T cell or beta cells, the cell therapy. So for the T cells typically is using for the cancer therapy, so such as CAR T cells, but the limitation for the CAR T cells typically is you need to get cells from the patient, but the patient can get limited cells. But if you can using like whatever cells resources come from, and then directly inject to the and modify it early and then inject back to the patient, it will be much quicker and directly open the market. So that is what happened for this one. The same for the beta cells, because you can like in vivo that culture, not the self that cells, and then directly inject back. And also because we have a long circulation for our the armored rebel cell, so we also can long circulation, like increase the circulation time for the nanoparticle therapy. So the nanoparticle itself typically is have like a short circulation, and if you increase the circulation property, it will help the nanoparticle typically go to the have higher chance to go to that specific site or targeting site in the so we do the animal, so it's animal for the mice. And also they can also for this we also have the cell sensor. This is help to resistant to the environment change. So even if in the very harsh condition, but the sensor, the signal is come from inside. So when our nanoparticle, that detector is only on the cell surface, so they can easily to detect the environment change. So this is our data. We can show they are kind of resistant of different that stimulation outside. So what we do is this is the regular, let me see. Oh, this is the nanoparticle. We're using the silver nanoparticle and give with different dose. So you can say when the dose increase, but this one is like a small, this one is a toxic. It's doxorubicin, so they kill the cancer because our exoskeleton have porous, so the small molecule can still go inside to kill the cells. So they will not block the communication between the cells and the outside environment. So the same thing is for this one. This is we're using the sugars which have no fluorescence, but when they uptake by the cells and they will metabolize and generate the fluorescence, so we can say they still can show the fluorescence. It means there is no effect for our metabolic of our cells. But for this is the nanoparticle. Silver nanoparticle is a 10 nanometer size, and due to the size that permeability, such selective permeability, so when we increase the toxic nanoparticle concentration to 12 microgram per ml, the supercells even can block them. So they were not physically attached, so they were not causing the toxicity for the cells. And also the same for the even larger one like bacteria, for example here. So this is we coating with our silica shells. So sorry, not silica shell, the amide that nanoparticle shells, so they cannot show the uptake of the bacteria. So the bacteria are not directly intact. So this is we're thinking they are helpful for our physically block the strands from outside. Also, we do is we see here, this is supercell and cells, we let it in the suspension, but this is cancer cell. So they have some like properties when they suspend for so long, so they will die during time. But as we see here, so supercells and cells, they have no difference compared to the cell viability during the times. And also we tried osmotic pressure, pH, different pH, and even the UV. So we finding our amide nanoparticle that coating the mammalian cells were not causing anything that will like decrease the death from all these stress. And this is for the delivery. So we're thinking we can preserve the original properties and then we can do the lung circulation. So this is the red cell. So we can see the red cell, they can still deliver the oxygen during the time. So they can have like we're loading the oxygen, release the oxygen and reloading. We can repeat several times. There is no effect for the red cell itself. But for the nanoparticle also the red cell itself can circulation inside that chicken embryo system. This is the capillary bed, and you can see this is amide red cells. They can go through here, even the small area of calibre of the animal stuff. So it means that the nanoparticle coating on the surface will not affect the circulation property of the red cell, since we know the red cell itself typically can circulate for so long. So we're thinking this also help for our nanoparticle delivery. So for this is our mice study. We can see, so for our nanoparticle, if we directly inject the nanoparticle itself directly, is only like 16 hours to circulation time. This is half-life of the circulation. But if we loading on the red cell, they can become 66 hours, which is at least 4 to 5 times compared to the nanoparticle itself. And also another thing is during our injection, the interesting thing is we can see in the lung. So they have some accumulation. This is also interesting because most of the nanoparticles, they don't have long accumulation that property. So our system is also benefit for the nanoparticle delivery in this case. And the next one is the multifunctionalization of our supercells. Since we can do different nanoparticle coating on the surface, we can also like loading, like using different nanoparticle to get different properties. For example, we can loading different color labeled of the MSN, the silicon misfolded silicon nanoparticle. So we can create a different color, like a multicolor that's a fluorescent nanoparticle. And also we can do the magnetic nanoparticle on surface, so we can control the strength of this one. This may be interesting for the cell therapy because you can control make it more accumulated in some area due to the magnetic field. And also this is what we're talking about sensing. So the interesting thing is we can show this is a show when the NO that released from the cells, the fluorescence will decrease during the time. So so we see at the first 8 hours, we directly can see the significance of the signal compared to the control. So which is this one typically when you're using the regular assay to testing this one, this need take around 24 hours to testing. So we have a more quick and early detect for this, sensing the cell release signals. And also we can release our nanoparticle shells on the surface. And this is our try to loading the the dye or cargoes into the nanoparticle, we're using the DAR1. This one can detect the NO as well in the blood. So we're using this one, we make the string for that one, we can detect the blood here. The blood concentration inside the blood, the NO concentration inside the blood. Also, we can do the same as magnetic control and the different colors. So right now we still try to do the different type of nanoparticle. We just show we can do it, but we didn't know if we can try more different type and combinations and what is the difference between each nanoparticle exoskeleton, because the nanoparticle itself is can differentiate different like amount of cells for us, and also we optimize the nanoparticle based exoskeleton to decrease the cell death and make sure they still have the same metabolic properties and all the things. In the future work, we will try the T cell therapy using this one, and also beta cell therapy, as well as the cell sensor. And the collaborator, Dr. Drew, is in the China right now, but we still do the collaboration for this one. Thank you.
Thank you, Jimin. Let's see, we have a question here. Not clear what your mouse study showed from a future patient benefit perspective. In a few sentences as possible, can you please state this? I guess you're asking about the clinical usage of these.
Yeah, so for this one, we have two. So my study have showed two things. This one is, my study is only show the nanoparticle delivery. So it means our armored red blood cell can help the nanoparticle delivery or lung circulation properties. But for the one which we're talking about, the cell therapy, such as T cell or beta cells, so this is, we're thinking because we can resistant from all this, all this different stuff on all these different strands. So we also thinking it can be help for the block the strands inside the animals, but we didn't really process the animal study right now. So but we will do it. So that is our future work.